Good afternoon and welcome to our Regeneron Science Talent Search webinar. I'm Allie Steeple, the director of the Regeneron Science Talent Search, and I'm joined by my colleague Elaine Edwards, who will give us a wave. Elaine and I together run the Science Talent Search, and we help to set the rules. We help with email customer service along with our team, and we plan the finals week and the judging, pretty much everything to do with STS. Elaine and I are the ladies behind it. And we're here today as the doing our third rules mini session to give you a little more insight into our rules for the competition and some things that are maybe commonly misconstrued or misunderstood, because we want to make sure that your project is actually eligible, and after you go to all that hard work of doing research and applying, that it doesn't get disqualified just because you forgot one little thing. So that's our goal with these three mini sessions. And with that, we'll move into our presentation today, which is about potentially hazardous biological agents and hazardous materials. If you're interested in our other rules to do with humans or vertebrate animals, we covered those in 2 other sessions you can find in our YouTube channel. Great. So if this is your first time tuning in for us to one of our webinars, and you're curious to learn what is the Science Talent Search, first, I'd refer you to go back a few presentations on our YouTube. You'll find a lot of overview presentations about the program itself, how it works, and how to apply. But just to give you a little bit of that overview today, if you're not familiar, the Science Talent Search is the nation's oldest and most prestigious science research competition in the nation. It's for 12th grade students who live in the United States or are US citizens who are living abroad. So in that way, it is a domestic competition, in contrast to our sister program, which is the Regeneron International Science and Engineering Fair. The goal of Regeneron STS is to identify future leaders in STEM. We do that through a holistic review process where we review your research project that you're submitting to us, an original project that you've conducted, and all kinds of other things about you. We're looking for those signs of potential to be a future leader in STEM. So your transcripts, test scores if you want them, activities, recommendations, all of that comes into play in our review process. Right now, the application is open. It opened on June 1st. I hope you've gotten started. If not, you still have plenty of time. The application is due on November 6th this year at 8 p.m. Eastern Time. Totally free to apply. If you haven't opened an application yet, open one today. That is something you might want to do if you're working with PHBAs or hazardous materials to learn a little bit more about how these rules we're going to talk about today apply to you.
So if you've watched any of our prior mini sessions, all of this on this slide is going to look a little familiar, but there are just a few reminders we like to start with before we dive into the topic of today's session. So as we noted, number 1 is that our eligibility and process, all information about that is available on our YouTube page and other webinars, but also on our website. We have plenty of resources for applicants and their supportive adults, some of which we'll highlight at the very end of this session, but plenty more that are available on our website. So if you are in doubt or in need of some assistance, plenty of things available to help you there. But these rules mini sessions are really designed to kind of deep dive into 3 aspects of our scientific rules. So we've had 2 sessions so far, one on our human participant projects, one about vertebrate animals, and today about potentially hazardous biological agents and hazardous materials. A couple reminders on all of these scientific rules areas, but especially true for PHBAs and hazardous materials. All entries to STS need to follow local, state, and federal guidelines. There are some rules that everyone needs to follow to keep yourselves and everybody else safe. We also know, however, that many labs and mentors might have their own sets of rules. A good reminder here is that Regeneron STS rules need to be followed if you are submitting your project to STS, even if our rules are stricter than those of the folks that you're working with. So just a reminder to make sure to screen our rules annually. We do some small changes every year, but that'll make sure that everything is in good shape when you submit your project to STS.
That's right. So like we've said, today we will focus on PHBAs and hazardous materials and the rules around them. We'll talk about what paperwork and pre-approvals that you might need and go through some examples. This should hopefully help you place the context of your research into the rules and understand better what you need to do. But just like in our last 2 sessions, Please understand that some of the examples that we give today may not perfectly match your exact scenario. We do encourage entrants to leave comments on the YouTube viewing today, and we'll follow up in a transparent way by next week so that people can see the answers to some of those questions. Or please feel free to email our team always at sts@societyforscience.org. We answer rules-related questions daily and are happy to help you with yours.
All right, so let's go ahead and dive in. Before you start any research project, we always say that it is a good idea to complete a risk assessment. You can find a sample of this on our website or in our rules book, and there is a copy here on the screen. This form is an opportunity for you to really articulate what you want to do, what safety procedures you're going to follow, and as an adult to supervise or sign off that what you're doing is okay and is safe. It is the responsibility of the student and of any adult involved in a PHBA and hazardous material related project to conduct and document a risk assessment. And that's going to help you define the potential level of harm, injury, or disease to any plants, animals, or humans that may occur as a result of your project. The risk assessment is really what determines what hazards and or biosafety levels are involved in your research. And that in turn is going to determine if your project can proceed, and if it can, what kind of lab facilities, equipment, training, or supervision are going to be required. Just to be very clear here, there are also certain types of research that are only permitted in certain types of environments. And so that's really going to help you identify what applies to you. If you want to work with anything hazardous or a PHBA, you're going to need to complete this risk assessment.
That's right. So by now you might be wondering, what is a PHBA? Or maybe you know, and maybe we'll give you a little more information about what counts, especially per our rules today. PHBA stands for Potentially Hazardous Biological Agents. You'll hear us abbreviate that throughout today's session. This includes microorganisms and recombinant DNA technology. So some examples are any kind of bacteria, algae, prions, fungi, parasites, several examples listed here. What sometimes students don't always realize is that many tissue or cell-related projects are going to count as a PHBA project. So anything we say here today, if you're working with a cell line, you'll need to adhere to these rules as well in terms of like filling out a risk assessment, getting a pre-approval, and just general safety procedures as well. So that's something to keep in mind. Research also involving human, so just to clarify even further, that also means any type of blood, breast milk, other bodily fluids that are coming from an established cell line or freshly collected, especially if from unknown origin. Those all count as PHBA studies. So that will dictate some level of location rules and safety that you need to follow.
So as we've mentioned, PHBAs are classified by biosafety levels, and those number 1 to 4. For Regeneron STS, our rule is that students may only work with organisms that are BSL-1 or BSL-2. So just to go through all of them really quickly, BSL-1 organisms, those are the ones that are considered low risk, and these can be handled in a high school lab. BSL-2 organisms, these are things that could make humans moderately sick, and these must be handled at an RRI, so regulated research institution, or a BSL-2 certified lab. It also exists in this hierarchy of BSL-3 organisms. These are ones that can cause serious disease, but these are prohibited for STS research. Likewise, BSL-4 organisms, these can cause untreatable disease, irreversible things like environmental impacts, and are prohibited. So just to kind of summarize this, you really need to work with a lab that is certified to handle your organism's biosafety level. So that BSL-1, you could do that at your high school lab, but at BSL-2, that's not going to be okay to study at a high school lab. You'd need to do that at a BSL-2 certified lab. How to find out what biosafety level your organism is, you can Google it. That's freely available information. You can also see page 27 of our rules book for some examples.
That's right. Even once you know the level of BSL or the biosafety level of your organism, there are some additional rules to keep in mind and some things even within that framework that are prohibited. or what we might count as a different level than you might think. So some of these special circumstances, first, let's talk about unknown bacteria. What does that mean? That's something that it has been collected from the wild. So you didn't purchase it, you don't know that it's coming from a clean place, scientific catalog sort of thing. But say if your bacteria that you're collecting, you don't know what it could turn out to be in terms of the biosafety level, that means that there are some rules around how we got to handle it, right? So like say, classic example, you're going to swab the water fountain at your high school. You expect some bacteria is going to grow on your petri dish. We don't know what kind of stuff you got going on at your high school water fountain, right? So the rule here is that if you keep the lid on that petri dish the whole time you're culturing it, you can keep this at a high school lab. You cannot take it at home, take it home ever. You should never be doing any of this type of research at home in terms of working with bacteria and anything that's a biosafety hazard. But if you are conducting this kind of unknown collection where you don't know how it's going to turn out, you can do this at the high school at a BSL-1 lab if those dishes remain sealed throughout the experiment and if they're disposed of via autoclaving or disinfection under the supervision of a mentor. In our rules book, you'll see a couple of times there are some instructions about how to safely dispose of bacteria. It's really important that you do that. You should not be opening plates. That's a really quick way to get a lot of people sick, very dangerous. Do not do that. If the Petri dish is open, then this is going to automatically count as a BSL-2 project, and it needs to have taken place in the RRI setting, a regulated research institute. And if we find that it is not, then that is grounds for disqualification. The second example we'll talk about is the culturing of human or animal waste. This is going to automatically be considered a BSL-2 study. So again, you'll have to do this at a lab that is biosafety level 2 or higher. And I guess a note, we didn't mention it in this session. We talk a lot about RRIs in the last session. What is an RRI? Every university college that has research labs, those count as RRIs, government labs. If you're not sure, you should ask. For the most part, this is an accreditation that we will be asking you about in the application, so you'll want to know where you worked. The next example, conversion. So if there is any chance, and as you research your organisms that you're working with, you should hopefully read about this. But if there's any chance that the BSL-1 agents could convert to BSL-2 during the course of the study, then the study must be completed in full at a BSL-2 lab. The next example, projects involving water samples from active harmful algae blooms are considered BSL-2. This is a relatively new rule for STS and for ISAF. A lot of students are interested in studying lakes, bodies of water near them that have these types of algae blooms that can really be very dangerous. So we are considering these BSL-2 now. Or they are BSL-2, you need to work with them in BSL-2 lab. Home environment. As mentioned before, you should not be doing any work with any microorganisms in a home setting. Certainly not culturing them. Anything like that is going to be disallowed. Prions are also prohibited. Any projects that are involved in prions are not allowed for STS. And same with anything that has antibiotic resistance. We don't want students perpetuating these things. Again, another way you can make a lot of things and people quite sick.
So in addition to these potentially hazardous biological agents rules, we also have rules about hazardous materials. What is a hazardous material? That's things like hazardous chemicals, devices, or radiation. Hazardous activities are going to be those that involve a level of risk above and beyond that which any normal person is going to encounter in daily life. So just some guidelines to follow here. An adult should be supervising all projects to determine if safety precautions are followed. This includes if you don't have a mentor, you should definitely think about what kind of adult is someone who can make sure and can testify later to us within your project recommendation that you followed appropriate safety measures for your project. If you are doing any kind of project with hazardous materials, you should sign off, you should have that adult sign off on a risk assessment prior to experimentation. You're also going to need to make sure that you are following all laws of your country, city, and state. We have a note here that this applies to drone projects. We know that these are becoming more and more popular too. There are definitely some differences between how different states and localities handle things like drones. But definitely, if you're working with any kind of hazardous material or engaging in anything that could be considered a hazardous activity, make sure you're doing that risk assessment and following all of the laws in your area.
Great. Now we're going to go through a few specific examples of different types of hazardous materials. This is an area, again, where sometimes some students don't necessarily recognize that what they're working with is a hazardous material. So like Elaine said, best practice really is to complete a risk assessment prior to starting your project. It does force you to think about, like, even if you think you don't need it, because you might find that you do, it forces you to think about, how am I going to approach things? How am I going to keep myself and others safe? Are there alternative methods to what I'm doing that could be better? It's also a reminder to do things like look at the material safety data sheets, which are provided by the vendor of any chemical that you're going to be working with. This is a great resource for, again, making sure that you're handling whatever you're handling safely. Are you in an appropriate environment? You better check, oh, I need to work in a lab that has a fume hood before I work on this. Can't do this in my kitchen. That's not safe. And STS will find out and will disqualify my entry, right? So we got to make sure we are safe just for ourselves too. So when it comes to chemicals, filling out that risk assessment is important for that reason, so that you're thinking ahead. You're going to assess the chemicals you're working with for toxicity, reactivity, flammability, and corrosiveness. The amount of exposure to a chemical must be considered as well. And you must include in your risk assessment proper disposal methods, and there'll be instructions for how to do that best within the MSDS.
So we also have hazardous devices and inventions. These devices can include anything that is dangerous equipment, really, in or outside of a laboratory setting, or things that are going to require a moderate to high level of expertise to ensure that you're using them safely. So includes things like high vacuum equipment, heated oil baths, NMR equipment, high temperature ovens, and so on. We recommend that all student-designed inventions as well have a documentation of a risk assessment. This is regardless of whether or not you're testing it on human participants, but especially if you are testing on human participants. Remember from our first session on anything human participant research, if you're testing an invention on anyone who isn't yourself, you're going to need some pre-approval from an IRB for that, and they will most likely want to see that documentation of your risk assessment, and they will help identify exactly what safety procedures you and your mentor, should you have one, need to follow.
Next, we'll talk about radiation. Again, if you're doing a risk assessment, which you should, you'll be asked to think about levels of radiation and how you're keeping yourself and others safe when using it. This is especially true when you're using radiation beyond what's normally encountered in everyday life. You'll see some examples here of especially what you need to think about. A note as well, if you are planning to use this project for ISEF, there are more specific rules within the ISEF rules around radiation. So we recommend that you take a look at those too.
For STS, we do permit research related to firearms and explosives, but we permit those when they're conducted, again, under the direct supervision of adult and when in compliance with all federal, state, and local laws. A couple of additional stipulations there. The use of the firearm must be conducted with proper state certification and training. And underage researchers are going to be prohibited from purchasing firearms, ammunition, black powder, explosives. So again, do really need to be working with an adult there. And students may also not build weapons themselves.
And certainly, again, as this should make total sense because this is everything you do must fit within local, state, federal laws, but certainly a lot of safety precautions here if you're working with those things. All right, prescription drugs and controlled substances. We do see students working with these materials, but again, this is where setting plays a very important role, as well as what you're doing and what you are using in your project. Students are prohibited from providing prescription drugs or controlled substances to humans or vertebrate animals. So that's not where you're going to focus your research if you are working with prescription drugs or controlled substances, but maybe you're working with plants or a cell line or something like that. You can use these things if you're working in an RRI. You cannot use them in a home setting, even if it's involving plants. The other thing that's going to be really important here, and you'll be asked in task 3 of our application to provide, is proof of the chain of command. How did you come to work with these materials? Were they prescribed by your mentor for the sake of this project? Were they materials that your lab has access to because it's a BSL or lab at an RRI, and this is what the lab does, is they work with whatever substance, and so you'll need to demonstrate that kind of paperwork in our application. So if you are curious about that, feel free to open one, go to task 3, answer the questions, and it will kind of give you information about what you would need to submit. The question we then also get often is, what about over-the-counter medications? You are allowed to work with these, but again, you can't give them to like humans or vertebrate animals. You can work with them in a home setting if you're working with like plants or something else.
So in all of this, we want to also take a minute to talk about some of the environmental considerations, things you should be thinking about if you're working with these materials for your project. It is the responsibility of the researcher and again, any adults involved in the project as well to protect the environment from harm. So that means we need to be thinking about what we're going to be doing with our materials once we're done with our projects. So just a reminder that the introduction or disposal of native or genetically altered or invasive species of any kind. So again, insects, plants, invertebrates, vertebrates, as well as pathogens or toxic chemicals or foreign substances. Putting any of those into the environment is prohibited. So again, need to be making sure that we're disposing of things properly at the end of experimentation and in accordance with their biosafety level. We also recommend that students reference their local, state, or national regulations or quarantine lists, again, just to make sure that you are in compliance with the laws of your area.
If you're not sure, what kind of safety I'm supposed to implement for my project, just complete a risk assessment as a first step, and a lot of the answers to your questions are probably found through that process. You should just be prioritizing safety and stop for yourself and any research subjects and the environment, as Zillian has mentioned. So when in doubt, seek guidance from a supportive adult. If you are working with a mentor, they're going to be the person who provides that to you. If you are not working with a mentor, but you want to work with something that's a hazard, make sure that the person who is signing off on your safety procedures is qualified to do so and knows at least a little bit about what you're working with, or can verify that you've read like the MSDS, that sort of thing. A good example of a project that there was some uncertainty around what they need to do to make sure that they're being safe to themselves and the environment. We had a project a few years ago where a student was working with a controlled burn. They wanted to light something on fire for whatever reason that was important for their project in an outdoor setting. And so they actually worked with their local fire department, had the fire chief sign off on their project that this burn would be supervised, in fact, at the fire station while the student did their research. They would be nearby, could respond very quickly if there was an incident, and that they'd given some advice to the student as well. So that's an example of a qualified person in that case who could make a call and help keep the student and environment safe. And also they could be aware, right, if there were any like burn notices in the area where it wouldn't be safe to do that project at all from an environmental perspective.
So obviously after this presentation, one of the primary ways that mentors provide support is just making sure that a student researcher is being safe and following the correct procedures for whatever materials that they are working with. But there are definitely a couple other ways that mentors can be really crucial in the lives and research process of their students. One of these ways is saving all paperwork related to the student project. In the application, students are asked to upload evidence of things like the risk assessment or other paperwork that they've done as part of their project. But mentors are too. Whoever completes that project recommendation has an opportunity to share everything they know about what the student has done, safety procedures they followed, and to share that paperwork. So if you save it, uploading it, just in case something goes wrong on the student end or they don't submit the right sheet, that can also really help your student. Also helpful is just starting your recommendations early. These aren't typical form letters, so they might take additional time to complete. Our system can be a little finicky, and so we just in general recommend that if you're providing a recommendation for a student, earlier is always best. You also have the opportunity to test our rules wizard in task 3 of our application. So you and the student should really be aware of what pre-approvals and paperwork might be required for a project. And a way to find that out really easily is by going through that rules wizard process in that task. It'll tell you exactly what you need. And finally, reading our STS rules annually. They do change slightly from year to year, so just a good idea to keep yourself up to date. Make sure that you're giving your students the most up-to-date information as well.
That's right. And just to add another point here, we are going to specifically ask the person filling out the project recommendation, did the student work with PHBAs or hazardous materials? And we're going to then ask you as the mentor or the supervisor, what safety precautions were followed, what trainings were taken. This is very, very helpful to have secondary confirmation, especially in cases, say, like paperwork was post-dated, that kind of thing, which is not okay for some types of things, but for this, as we'll talk about some of our examples here in a minute, we do see from time to time. But if the mentor, in the case of STS, is verifying in the recommendation form, yes, all of this happened before the experiment took place, and this is what we did to keep everybody safe, then we tend to go with what the mentor has said, and then the project is okay. So let's go through some common scenarios. Oops, did it up a little fast. Okay, like we said before, these scenarios may not perfectly align with your project, but we hope that going through some examples will provide some clarity for some people. So the first example here of something a student might ask, can I monitor my Petri dishes in my home lab? No, you cannot. PHBA research is prohibited in a home environment. You would have to do this depending on the BSL level of the organism you are working with. You would need to do this at your high school or at an RRI.
All right, next here is what if I didn't complete a risk assessment before I started? So this is a little of what we were just referring to. This is okay with some, again, stipulations. So one, we'll want to see documentation of a safety process that you did follow. So this is, again, a case where it's really crucial for a mentor to be involved and in that project recommendation to be very clear about what safety process you followed if a risk assessment was not completed in advance. You can also fill a risk assessment out after you have completed your experimentation. But again, we're going to want to see that verification from the mentor that that is in fact true. It's not just a form that you whipped up when you realized you needed one in your application.
That's right. And the other point to make here is we also understand that your lab might have had you complete different safety forms than ours, right? That ask different questions. And so that is acceptable paperwork as well. Anything to demonstrate that you did think through this and that everybody was safe in the process. What if my mentor works with a BSL-4 organism? Or more specifically, I think a later example, my mentor is working with the BSL-4 organism and is giving me data. Is that allowed? If you recall, BSL-4 organisms are bad news. You are prohibited from working with them directly as a student entering Regeneron STS. That is grounds for disqualification. However, the loophole is if you have a mentor who is working with this and they want to give you data about their study, images, that kind of thing, and it is very clear everyone is on the same page that and is letting us know, confirming, verifying. Student did not ever touch any of this. They were never allowed in here. A lot of these labs are not going to let a high school student in, to be fair, if you're working with something like that. It depends on the organization, but you can use data that is given to you. And if your mentor allows you to design an experiment, if that's something that your lab is okay with and they would like to implement it for you and you do not touch or get near any of it at any time, again, if all of that is super clear, then that is okay for STS.
I would like to test out a few over-the-counter medications on my cat. Not okay. No, this project would absolutely fail to qualify. Please leave Fluffy out of your experiments.
Right, big violation of those vertebrate animal rules for sure. I used E. coli to feed my C. elegans, my nematodes. Do I need a risk assessment? So if you used a bacteria to feed your nematodes, yes, you should fill out a risk assessment if you're working with bacteria. The other thing to note is that this one does trip students up because they don't always think about these C. elegans projects as PHBA studies. But if, when you're doing your risk assessment or like planning out your project, a good way to think about it is like, what am I using? Right? Like what chemicals, bacteria, whatever, like what food source, all the things, list them out. What are they? How safe are they? Check the BSL level, check their MSDS. This does count as a PHBA study. Depending on what you're feeding to what, it could very well be okay to do at a high school. Most strains of E. coli, and you should check our rules book, but most are going to be okay to do at a high school. They're considered BSL-1. And then sometimes there might be a food source that's BSL-2. I don't know. It depends what you're doing. So do check that. That's where that specificity comes into play.
All right, next one. I worked with blood from a blood bank. This is something we've touched on in both of our previous rules webinars. This is permitted. It would be classified as a cell tissue project. Because it's publicly available, it's also exempt from any kind of pre-approval, but we would want to see documentation from the supplier. So again, just evidence of where you got this tissue and that you had permission to use it. We'd also probably want to see a risk assessment for this.
And then the last one we'll go through, I worked with the BSL-2 organism at my high school. Well, most of the time, this is not going to be allowed. Most high schools are considered BSL-1 for their biosafety level. However, I do know of a few, and I do mean a few, like count on one hand, high schools in the United States that have BSL-2 labs. Ask your teacher. There are some specific requirements you can find in our rules book, like what counts as a BSL-2 lab, like in terms of what like equipment and safety equipment that they need to have on site. And it is unlikely, most high schools are not BSL-2. If yours is, and you're very lucky that yours is, then you can do that type of work at that high school. Your teacher, mentor will need to like sign off, like, yeah, our high school actually is BSL-2. We have a little list that we keep as well, so we're likely to know depending where you're doing this. But yeah, it's rare, but it is, we do see it.
All right, so just to kind of wrap it all up here, like we said, you have plenty of resources available to help you in addition to this webinar. We have a couple of documents on our website. One of them is our common FTQs, so our fail to qualify document. We post every year a list of the top reasons that students failed to qualify in a previous year, just to help entering students kind of get a sense of things that have tripped students up in the past and make sure that they don't fall prey to those in their own projects. So definitely take a look at that. Of course, you'll also want to take a look at our full rules book. We know this is a very long document and it is updated every year, but it does absolutely have everything you need in order to make sure that you are getting the proper pre-approvals for your project, that you have the right paperwork, and that you're keeping yourself and others safe. Um, if you haven't started your application yet, we would hope that you do so immediately after watching this webinar. Always better to get started early, especially with the new school year around the corner. So visit sciencetalentsearch.smapply.org to start your application today. If any supportive adults are watching, you are also, of course, welcome to start a test application. In task 1, you're able to indicate that you're an adult testing, just so we leave you out of our official counts. As always, if you do have any additional questions that aren't answered here or on any of our websites, you can contact us at sts@societyforscience.org. Our staff man this pretty much 24/7, and we are always happy to help students or supportive adults with anything you might need. Final reminder here that the deadline for our application is November 6th of this year at 8 p.m. Eastern Time. So that includes the entire application. That also includes recommendations. So all materials need to be in by that deadline. We will hope to see you if you are watching this now. We will hope to see you in the entrant pool this year.
That's right. Thanks for joining. Good luck. Bye-bye.
And we're done.
All right.